All right, welcome everyone. Thanks for coming to today's Tools and Technology Seminar Series. I think we've got some new faces at the beginning for this being the first one of the semester. So for those who are not familiar with the seminar series, it is a venue to discuss tools, technologies, methodologies. Sometimes these are recently published, but they don't have to be. Sometimes they're currently in development. And sometimes they're things that have been out here for a long time and are just being used by researchers, and someone in the community thinks that others may be interested in knowing how they're used or that they exist. So keep that in mind. I'm always looking for future presenters. We do have an online audience. So if you're online and you have a question, you can raise your, you can put it in the Q&A pod, or if you really want to ask it verbally, I think you can indicate to us that you would like to do that and we can unmute you. If you're in the room and you got a question, of course, just raise your hand like you normally would. Please try to speak up so that the camera, or the microphone can pick you up your question for the online audience. So with that, I would like to introduce our speaker today. We have Kai Wang, who is a postdoc in Maureen Sartor's lab in DCMB.
Okay, thank you. Yeah. It's a great chance for me to talk about the project I'm currently working on. So today I'm going to talk about the title of my talk is like the gene target prediction of environmental chemical using the coupled matrix completion algorithm. Yes. And so for environmental exposures contains like a different type of like a chemical, physical, biological. But for here, most of we only focus on chemical exposures. And based on a news, there's like every year there's like 2,000 new chemicals will be released to environment. And some of the chemicals is not good to our health. And so based on this news, it's like every day we have 7 new chemicals released to our environment. So to do a comprehensive toxicology assessment of the new chemicals is impossible. Even in the PubChem, they already have over 1 million chemicals registered in that database. But the correlated the biological data is only around like only 10% of them have like a biological data. So which is like, yeah. And then so traditionally we use like a lot of like in vitro or in vivo toxicity testings. So in vitro, which is like we use a lot of cell lines to do to add in different concentration of the chemicals and to testing how the cell works. But the hard part is like for this type of method is like it's hard to find a right cell type organelle to mimic the real like in vivo environment. And in vivo, which is like we use a lot in the SPH, we use a lot of mouse to do the study. But the other problem for this method is like it's very expensive and time consuming. And also human and mouse are different. Yeah, so it cannot, and also those are like cute animals. So it's hard, yeah. So yeah, so in US actually, the EPA and FDA, they proposed this like a toxicity testing in the 21st century. So which is like based on this figure, you can see in 2042, so all of those like personal care stuff, all those chemicals should be only used like the computational way or in vitro way to testing the toxicity of the chemicals. Yeah, so this is in US. And actually in Europe, they already set a law that is all the personal care stuff. You can only use computational way to testing the chemical used in personal care products. And I met a guy in the PNG. So that guy actually is doing those type of psychology computational stuff in PNG. And he told me like it's like in Europe, because they want to sell the products in Europe. So they can only use any like biological based data to support the idea the product is not like a toxic to human if that paper published before 2011. So any paper published after 2011, they cannot use that even as a reference to support, okay, this chemical is safe to human. Yeah, and also in the EPA, I have a figure here. So in EPA, they have another way to do the toxicity screening is like high throughput toxicity screening stuff. So they have a team to do that. Is like they have a robot arm. They have like 4 teams. So the first team is like only to prepare all the cells and chemicals, whatever. They have like a huge like a rack to put all the stuff out there. Then they have a robot arm to do the experiment like every day and not stopping. So in that way, they can testing like 1,000 chemical in like 2 weeks to do, but it's still like not fast enough to testing all the chemicals. So that's why we need to use like some computational method to do the toxicology research. So the first one, so this is like the very most widely used method. It's called Medial cross. So this method is like so the logic is very simple. So similar chemicals, they probably have similar biological activities. So for this method, so they just what they did is like they just compare the chemical structure. So for example, if this one, so we don't have the biological data for this chemical, but we have like a biological data for those 3 chemicals. Then they just compare the chemical structure to say, okay, because those chemicals are very similar to each other, so this chemical in here probably have the similar biological activities. So this is very fast. So the model of this method is like very easy to interpret it. But the problem is like they don't include any like biological data here. So everything is just based on the chemical structure, then to give a guess of the biological activities. So then after that, so this method is developed called the structure activity relationship, or the quantitative structure activity relationship. Yeah, for this one, it's like a different from the real cross. For this one, the model will include the structure of the structure of the chemical and also the biological data here. So then use those two type of different informations. Then they can train a model to like predict the toxicity of the chemical. Then when you have a new chemical, you can just use a structure, then get the like the numerical features of the chemical, then to predict the biological activities. And so for the QSAR methods, you can use like a different type of machine learning algorithm to do this. But for all of those methods, the limitation is like most of the time they only checking whether this chemical can activate a marker gene or a certain marker gene or certain pathways, which is like a binary classification problem. They cannot give like an overview of the biological activities. So then for my study, we use like the matrix completion method to do this. So for matrix completion method, which is like basically it's like imputing the missing values in a partially observed matrix. Yeah, so in a drug, so this type of method is actually very widely used in drug repurposing and repositioning. And usually for the drug target interaction, so they use binary numbers, and usually they just predict the physical interaction between the chemical, the drug, and the proteins. But for environmental chemicals, so they probably have like a different type of interactions. And so a traditional way to do a matrix completion is called the SVD, singular value decomposition. So usually, so basically it's something like you have a partially observed matrix like here. Then you can break down this matrix into 3 other matrices here. Then when you multiply them together, you can have a new matrix. So then when you do this, you can initialize some values in those 3 new matrices. And then you can adjust the values to make sure the existing value in the A can be recalculated after your modification of those 3 matrices. Then you can find in like a kind of like a missing values. But you can see from here, those part here and here, is actually not used in the calculation. That's why they have like the reduced SVD, which is like remove those two parts because there's not used for the calculation. And after this, they have another like a truncated SVD, which is you can see the dimension here is different. So basically just brings the size of the matrix because a smaller matrix you can use like less time to do the calculation. Otherwise, it would be spend a lot of time. So a famous it's kind of like a famous problem that is fixed solved by the matrix decomposition is this one. It's called a Netflix problem. So this is like I think it's like more than 10 years ago. They have Netflix. They have this competition to ask people to develop an algorithm to solve the problem. So the problem is like they want to recommend movies to different users, but not all the users will rent all the movies in the system. So that's why they have like a partially observed matrix. Then this type of method is called the func SVD. So because it only break down the matrix into two matrices, and then you can initialize some values here and here. Then you can, when you multiply them together, you just to make sure that the existing value in this matrix can be recalculated. Then the other missing values can be used to do the recommendation movies to other users. Yeah, but in a real problem, so the features, so those two breakdown matrices probably do not have real meanings like the coordinates in PCA. And so for here, so we actually have a new algorithm. It's called the coupled matrix completion. So the traditional way is like you only use this partial matrix, which is A here, to break down into two matrices, then do the calculation. But for here, what we did is like besides this one, so we will also have two other coupled matrices. So for example, for the drug target prediction, what we have here is in the A matrix here is like the row probably can be the different drugs and the column can be different proteins. And then the matrix C here is actually the similarity between the genes, and the R here is actually the similarity between the drugs. So all the hypothesis of this method is like a similar chemical probably have similar targets. Yeah, that's the hypothesis. So the method for here is just like all of those three information matrices here will be used to regenerate the three matrices. Then multiple those three matrices together, we can have a new matrix that have most of the missing value is imputed. And yeah, so this method is actually developed for drug target interaction prediction. And the paper is actually published. Yeah, it's published like in 2020. And in drug target prediction, because drug is actually designed to target a specific protein. That's why usually they use a binary interaction matrix. So that matrix A have just 0 or 1 to do kind of like a binary classification. But for those environmental chemicals, none of them are actually designed to target human gene. They are just use the products. So that's why we think a binary number probably not good for this type of prediction. So that's why we use a non-binary value here. So the algorithm will look like this. So the input will be have like a three matrices. So for our study, so the main matrix, which is MEG here, it's a chemical gene interaction matrix. The number in there is not binary. It's just the a decimal number there. And then the two coupled matrices is the chemical similarity and the gene similarity. And so basically it's like the algorithm is something like we need to pre-process the two coupled matrices. It's kind of like the normalization. So for this step, we for each of the matrix here, we will have like a two hyperparameters to adjust. And then we will grab the existing value in this matrix and the corresponding position of the existing value in this MEG matrix. Then initialize like two B1, B2. So the B1, B2 actually have the same size of the MGG and MEB. Then we run this iteration, and then we can regenerate the final matrix by updating the B1, B2 numbers here. And a good way for the good thing for this algorithm is like it converges very fast. So for our data, we only run the iteration like a iteration number is 2. So it's like we only update the B1, B2 like 2 times and convert finding the best output. And the data we actually use for our study is from this database called the CTD database. It's the comparative toxicogenomics database. So this is the best database that have the environmental chemicals and as related biological activities. And they contain like a different species, like a mouse data, rat data, human data, or zebrafish data. But for us, we only grab the human data. And this data set is actually, those numbers here is actually downloaded 3 years ago. So if you download new data, the number probably different, a little bit different. Yeah. And for this data, so after we filled out some drugs, so they have like almost 5,000 chemical exposures and like 22,000 genes. And the existing interaction number is only this. So if we put into a huge matrix, it's only like less than 1 point of the huge matrix is actually existing here, the interaction matter. So that's why if we want to do a matrix decomposition on this type of matrix, because this is too sparse, so the number, if you just only use the value in here, then to do the decomposition, so that it's hard to control the results because it's too sparse. Yeah. Yeah, so this is the interaction matrix. Yeah, so for this data, we actually have we calculate the interaction between the chemical and the genes in 2 ways, and then we only use the first way. So the first way is like we just count how many gene and chemical pairs are in this database. Because a different publication, they probably test the chemical and the gene on different level. Probably one publication is on testing the chemical on RNA level, one is on DNA level. So we didn't actually differentiate those different type of interactions. We just count how many times of the chemical and genes they have a interaction in the literature. And the second way we count is like we count for each of chemical gene pair, we count how many unique publications for there. But the publication number is not nice. It's not performance not good. So we only focus on the chemical gene pairs, and then normalize that count into a number between the range 0 to 1, then to do the calculation. That's for the interaction values. And then for the chemical similarities, we only use the RDK, which is a Python package, to calculate the 2 type of the fingerprints. One is the Morgan fingerprints, one is like a topology Torsen fingerprints. Those are like a very a normal way to do the chemical similarity. Then we calculate the chemical similarity based on those 2 fingerprints. And those 2 figures actually the distribution of the chemical similarities of all those chemicals used in our study. You can see it's very towards to 0, which means it's like even for those 2 coupled matrices, it's also have some missing values because some chemicals cannot have like finding a good similarity numbers. And for the gene similarity here, so we use an R package called a GoSamSam. So this package is actually calculating the gene similarity based on the Go term the gene assigned. So because there's like 3 type of the Go terms, so we can calculate 3 type of the gene similarities. I think GoBP is very well annotated, so that's why you can see a lot of numbers here. But for other 2, GoMF and GoCC, they have a lot of a high bar in the 1 here. Yeah. So yeah, so those are the data we have. So we have the interaction matrix and 2 type of chemical similarity and 3 type of the gene similarities. And then we can just run our method to predict to test the performance. So for here, we actually compared our method to other 3 similar matrix completion algorithm. So all of those 3 methods is implemented in MATLAB. And we chose those 3 because those 3 also need the interaction matrix and also the 2 coupled matrices. But the difference between our method to their method is like so the 2 coupled matrices, the value from those coupled matrices in our method will be directly used into the imputations. But for other 3 methods, they only use those 2 coupled matrices to adjust the value in the interaction matrix, then do a matrix decomposition. And yeah, to compare those three methods, so in their paper, in this paper, CMF paper, they also mentioned a pre-process algorithm, which is called the WKNKN. So this method can improve a little bit the performance of those three methods. So in that way, so we actually compared our method with these three methods and with and without the WKNKN. So it's kind of like compared to like 6 different methods. And we defined some like a true positive and true negative interactions from the data set. And then we just calculate the AUC to compare the performance. And to get the true positive and true negative data set here, so for the true positive data set, what we did is like we choose the top 20 most well-studied environmental chemicals. And then we choose the corresponding target genes, the most well-studied target genes of them. Use those as a true positive. And for the true negative, we also use those like a top, most well-studied top 20 genes, and then use those genes and never being reported as a target gene of those chemicals as a true negative. So the interaction matrix was something like this. And then when we actually calculate the AUC, what we did is like we just remove some like existing values, and then we use those values to repredict those values, then calculate the AUC of the method, and to compare the performance. Yeah, so here, for the performance comparison, we actually generated 3 different size of the matrix. So it's like a 200 by 400, 400 by 600, and 600 by 800. And the first 3 row, for the other 3 methods, it's like a without the WKNKN. And for here, it's like a with the WKNKN. And you can see with the WKNKN, it can improve the performance of other 3 methods. And when the size of the matrix like increasing, you can clearly see that the performance of other 3 methods is actually getting decreased a little bit. But for our method, it's very stable across different sizes of the matrix. And also, this is the runtime for our method, because other 3 methods actually in my lab, so it's kind of slow. So when they actually run the 200 by 400, they probably need like 5 minutes to run. But for our method, I implemented in the method in C++, so it's very fast. It's only like several seconds. So that's why when I do the case study, I run 200 different data sets. It only took like 20 minutes for all of them. And after that, we performed a case study. So when we run a case study, on 5 different class of chemicals. So the first one is BPA and its analog. So BPA is actually the chemical used to make those plastics transparent. All of those plastic like cups. They probably, for now, BPA is already banned in US, but they probably not have BPA, but they also have other similar chemicals. Yeah. So if you buy the bottled water from Costco, you can see their label non-BPA, but they probably have other like similar chemicals. And the second one is PFAS. So PFAS is like a very a huge. They have hundreds of PFAS there, and those chemicals are actually used in those non-sticky pan. So if you go to Target or Walmart, some of the pan they will say non-PFOS, because PFOS and PFOA is like the most like a very well studied. But they probably have other similar chemicals. That's why it's not sticky pan. And then the third group is called the docking. So the most famous one is called TCDD. So TCDD is actually during the Vietnam War. So the U.S. Army used a lot of chemical called Agent Orange, so which is like a herbicide to kill the grass. But the TCDD is actually a byproduct of the Agent Orange. But when they actually use this chemical, they don't know TCDD is there. So after they found TCDD is there, so they already use a lot of those chemicals. All the soldiers and the people in Vietnam, they already exposed to this chemical. And this chemical is actually very bad to human. They can cause all type of cancers and stuff. So yeah, so we use like PCB and other similar chemicals for the third group. And then we use PCB. PCB is actually a chemical also like banned in the US like many years ago. In 1979, PCB is banned in US, but in some old like electricity devices, those still have these chemicals. And the last group is those benzene and methanol, which is like used to keep those biospecimen. Yeah, those two chemicals. And for the case study, because the ultimate goal of this research is to finding the potential target genes of a chemical without any biological activity data. So when we do the case study for all of those chemicals, what we did is like we actually removed all existing interaction values for each of those chemicals from this like a huge matrix here. And then we break down this matrix into like a 200 data set with randomly selected chemical and genes. For each of those small matrices, it contains the case study chemical and all the existing value is removed. Then we run the algorithm on all of those 200 different sets. And then we can impute in the target genes for all of those in all of those 200 data set. So in this way, each chemical gene pairs can be imputed like 10 times averagely. Because if you use this one, it's not working because it's too huge. We will spend a lot of time to do the calculation. But for each of those here, the size is 300 chemical and 500 genes. So it only took like 20 minutes to run all of them, and then we can find the results. So for here, for the case study, so for each of the chemical, we generate a set 20 different 200 data sets for them, and then we run them. Then we can grab all the imputation values. Then we compare our results to two different data sets. So because all our data is actually from CTD, but when we do the case study, we remove all the existing value. So then we compare our imputed value to the existing value in the CTD to see how it looks. So from this figure, because even in CTD, not all those case study chemical, they have a lot of like target genes in there. So we only compared those like 6 chemicals. They have like a whole bunch of like target genes in CTD. So in here, we actually, because our algorithm will predict like a number, it's like a bigger number means the chemical have a higher possibility can interact with that gene. So we just order the results, all of the CMFC, our results, and then we can check how many top target genes from our calculation is actually also a top target gene in CTD. So in here, actually, it's like for the BPA, this is like the very well-studied chemical. We found 83, I think it's 83 chemical genes. It's in our top 100 gene. It's also in those genes is also like a top 100 gene of the BPA in the CTD data. So which means like our algorithm can predict the target genes of those chemicals. And after the CTD comparison, we also compared our result to the CompTox database. So this is like the EPA generate this database. So in this database, it contains all the data is actually based on the biological, like in vitro assays. It's like the high throughput something. And then in this database, also they only have like this 11 chemicals. They have biological data on the CompTox. And then we compared them to a similar comparison to see how many target genes actually predicted in our result is also target genes in the CompTox. So for here, you can see those numbers here is something like in CompTox, they only tested like they only have like 54 target genes of BPA. And our result can predict 20 of them as a top 100 target genes for BPA. So those are numbers, I mean, yeah. And after that, we also checked the AC50 from the CompTox, because from in the CompTox they have the bioassay data, so they have the AC50. So the AC50 in toxicology is like the concentration at which 50% of the maximum activity of that gene is observed. And so in our result, a bigger number means high possibility the target gene, the target chemical can interact with each other. And for the AC50, it's like a lower AC50 means that that gene is actually very sensitive to this chemical. So in that way, for all of those genes like included in our study, also in the CompTox, what we did is like we just use the top 33% as a of the AC50, that's a low AC50. You just order everything based on the AC50 and order our prediction result, and then we compare. So if in our result, if they have like a higher imputed value, and then we check if those AC50 is like a lower, so which means in here something like that. Yeah. So from here you can see mostly it's like they have this pattern, like our result have a higher value, which also have a lower AC50 in the CompTox, so which is like a very nice result. But in our calculation, we didn't have any AC50 information in our data, so this is actually a surprising result. We didn't expect this, but actually for some of the chemicals, they are like matching with each other very well. And after this, we also compared the pathways. So pathways of those target genes from our result and also the CTD data targets genes. So in here, what we use is like because we only use the chemicals that have more than 100 significant gold term enriched with CTD target genes in this comparison. And for our method, we use the top 1,000 target genes from our prediction, then run the David to find the enriched gold term. Then we do a correlation between the results to see if the target gene from our result can be used to finding the similar enriched the gold terms from the real data. So you can see for I think only for this one, so the correlation is not significant. But for other chemicals, so they have like the significant correlations between each other. So which means that our method the gene that predict from our method can also be used to finding the pathway related to those chemicals. And then we zoom into the BPA, because BPA is like a hormone related chemical. So for here, we actually grab all the hormone related gold terms from our results and to see. So because those four chemicals, you can see the structure, they are very similar to each other. So we expect that those gold terms will be like very similar to each other, but not exactly the same. So we can see, so most of the gold terms of hormone related gold terms is actually enriched for all of those four chemicals, but there are some like a difference here. And then here, this comparison is actually we overlapped with our significant gold terms of those four chemicals and also the significant gold terms from the CTD database. Then we found the overlap ones. So all the overlap gold terms of all of those chemicals is like a hormone related. Yeah, so which means that our prediction result is not like a go away for other like a weird result. So this is like our result can be used to find like a significant gold terms from those genes. And then there's another like a result. It's like for this gene, GPR1 gene. So in our prediction result, we only found this gene as a top target gene of the BPB chemical. And then we found this paper. So in this paper, the authors found that the BPB actually have a 9-fold binding affinity than BPA to this gene. But in our result, we also have found this gene as a target gene, a top target gene of BPB. So which is so this paper, the result from this paper kind of like support our results. Yeah. So then So that's all for the work for this talk. And then for the future plans, because everything for now it's CMMC, which is coupled matrix completion. So even though we have like a different type of the gene similarity and the chemical similarity, when we actually do the calculation, we only use 3 matrices to do the calculation. For the second step here is like we can use like all different type of the chemical similarity and the gene similarity together as a tensor, so which is like a 3D matrix to do the coupled tensor matrix completion. And then after that, the third step is actually to do a coupled tensor completion. So for here, this tensor actually can be Because for now, everything is just the gene chemical interactions counts there. But for in future, we can add in the actual diagonal here to have like probably we can specify the different target tissues. We can generate a different target tissue specific interaction matrix. Then put them into a tensor, then to do a tensor completion. Yeah. Yeah, basically that's all. And then so yeah, so this acknowledgement is like my three mentor, Maureen, Dina, and Justin, and all the co-authors of this paper. And this project is supported by my K99 grant. Yeah, that's all. Yeah.
Thank you for your nice presentation. I'm wondering that if you develop your current model TMMC to CTMC or CTTC, then can you handle the isomerism well? Because most present QSAR models are not handling isomerism well. So
You mean isoform, right?
yeah, isoform, yeah, that's right.
For the gene isoform?
And so I mean, even the structurally similar chemicals can have very different biological activity because of the chiral centers. So, I mean, is your model handling the chirality or even in the future direction?
For now, that part of information probably can be used to generate the chemical similarities. Yeah. So after the chemical similarity is fixed, so those algorithm is actually not really take that part into the calculation. Yeah. Yeah. Yeah.
It's a full term as your evaluation matrix.
A similarity matrix. Yeah, yeah, yeah.
So from some full terms you have the opposite function. Some is positive regulation, some is negative regulation. Figure out. So when you use the full term, you will consider this probably for one chemical and the protein interactions. You have an opposite function.
No, for here, no. For here, we only use that package to, actually, it's not using the goal term. You mean the similarity matrix, right?
Yeah.
So for that package is only calculating the gene similarities based on how the goal term is assigned. It's not actually have the goal term information. For the data we have there is like for gene A and gene B, there is a similarity number.
Okay.
Yeah.
So the genes you use the goal term.
Yes, yes.
See if they have more overlap, goal terms they will more.
Yeah, they probably have like a similar. Those two genes probably have more similar.
Okay.
Yeah, yeah.
There was a patient data, right? The early sec of the transcription for the matrix.
I'm sorry, which matrix?
Sorry, to know how much are the gene expression for the matrix, that was from patient data?
Oh, there's no expression data. No expression data. For here,
What about the patient, how it affects the genes? How do you?
yeah, so the in the action is like it's just from the database CTD. So there's no expression data.
Okay.
Yeah, we just count how many chemical gene pairs are actually in that database.
Okay.
Yeah. So there's no expression. Yeah, no expression. Just counts the how many. Yeah. C++. Yeah. I also implemented this method in Python, but with NumPy and pandas, but it's very slow. Yeah, it's like it works, but yeah, but the Python is very slow, so that's why I changed to C++. But now I'm developing a R based this method. I'm developing an R package for this. But the core confirmation calculation is still in C++. I use RCPP to do that. Yeah. For the RCPP version, for pure C++, for a smaller, like 200 by 400, it's like 3 seconds to the calculation. But for the RCPP version, 200 for 400 matrix, they probably took like 10 seconds. Yeah, for the largest one for here is 10 seconds, but for RCPP it's like 30 seconds. But it's R, so that's why the original algorithm is actually in MATLAB. That is implemented for the drug target prediction. But I'm not a MATLAB guy, so I don't know how to MATLAB.
Will that calculation be difficult of metric multiplications? Because could you offload it to a GPU or even faster processing?
Probably yes, but I haven't tried using GPU. Yeah, probably yeah.
I should have another few seconds then.
No other questions, and we can kind of run.
Okay.
Thank you all for coming, and hopefully see everyone next week.
I know.